Hello everyone. So as a part of the wavelet analysis using the VEPs, I was giving a presentation regarding that. So based on that, I'll just give you a small demonstration of how to do the wavelet analysis. So first, as usual, we will do the usual EEG analysis. We just open the EEG lab. So here I am going to use a VEP data. So I'll just import the CNT file. We have been using the CNT file for this VEP. So I'll just take a VEP data. So here we get the. So I just open this VEP demo. Over here also it's a CNT file, so there are 69 channels, and here the events are just 120 because there are only 120 events and triggers we have been giving, and here the sampling rate is 1000. So here this is the place where we have to do the sampling. If we have to do the same procedures for the other data also, we have to change the sampling rate. So since I'm just showing example with the VEP, I'm doing this sampling rate change for this particular thing. So we just make it 1024 because this is a power of 2 also, and we have to obtain this epochs in that range also. So that's why we will take 1024. So in this case what happens is we will have the sampling rate change to, so it gets resampled and we will have 1024. So here there is a resampled data. Before doing any step, we have to add the channel locations. So we do the usual step of putting the channels. If it was any other data, they would have the channel locations inside the channel, I mean in the continuous data itself. So here we have to externally add the default channel locations. So this is how it has been done. So here we have the channel locations being included, and after this I just give you a pre-filtering step we have to do, that is the notch filter. So we just do the notch filter. So the notch filter, it will get filtered. So based on that filter, we can, after that I'll show you how the triggers look in the VEP. So this is the, we'll do the scroll data analysis over here. So over here these are the initial responses which are having a lot of artifacts. So I'll just change the display to 30. So here the red ones, all these red ones are the data, the triggers, the VEP triggers that has been obtained. So here only one trigger option is there. So this is about the VEP. So here we have to create this alone, this one's event list we have to create. So first we have to do that process of creating the EEG event list. So here we use an advanced option, and here we load accordingly that. So this is the VEP. So here the first one indicates the trigger name, that is the one is the event code, the event type which was there. We label it as VEP, and then we put a bin number as 1, and then we do a checkerboard. The bin description is that it is a checkerboard. So this is the event list, and we just apply. We just have to enter it over here and update line, so it will get added, and you can just save it for future purposes also, and then you just apply it. So what happens is all these event which was named as 1, it will be now named as bean 1, or it will be named as checkerboard, something like that. So I'll just show you how it has been changed now. So followed by this, there should be a epoching that has been done. So here all of them are being named as VEPs now. All the ones are being changed to VEP. So this is why we do the event list options in order to make the program understand that these ones are nothing but our triggers of our interest. So next is the bin epoch. So epoching is to be done. So now what has happened is we have to be very precise about this now in this case, and for wavelet we have to consider this frames per epoch also, because we want the frames per epoch also to be in the range of in the powers of 2. So since we have given this 1024, what happens is we will get the epochs. If you do from minus 1000 to 1000, what happens is this whole thing will be given in that particular range. In 1-2 seconds we will have this particular. Now what happens is we wanted only a particular part of interest, that is just till 200 maximum, not more than that. So what happens is we just want the first quadrant. So first 25% of the sample is only we are taking. So we take from minus 50 to 200, so that we get the 25% off to this 1204. So it will be there, we will get a frames per epoch. That means for one epoch we will have 256 data points. For that reason only we are doing the epoching in this range. Okay, so if you have wanted in P300, means we will take from minus 50 to 700, so that we will get in that range, and also we will get in the powers of 2 as well. So that is why we take like a first quadrant kind of thing we will take in this section. So if we run it, what happens is now all these we will have 120 epochs. Along with that, each epochs will have 256 data points. So this is why we did that particular epoch range. So if we suppose if we do a different for example, now if I change the epoch and I am just doing it like I'll go back to this event list after creating event list. So now I am doing the bin based epoch. Now what I do is I just don't now if I don't do this particular range, for example I am doing something like 300 for example. So now here we have 350 data points will be coming. So okay, so if I just run in this manner, what happens is we won't get the powers of 2 in here in the frames per epoch. So it will be changed, it will become like say 358 it has become. So we don't want to be in this range, we want always to be in the powers of 2. So for that reason only we are doing it in this as I mentioned over here, I have done in this case. So this is just I gave a demo that why we have to do from minus 50 to 200, why I just explained over here. So so that we get up this 2 to the power n in this range, we get our data point. Okay, data sets we get. So this is about the event epoching. Next we have to do is the usual step, that is the artifact detection, that is also voltage threshold. Even this the time period of our experiment time period is less only, but still we have to remove all our artifacts. So here we take this is a 64 channel, so I am just removing all the trigger and EMG, HEO, VEO channels and everything. So and then we accept it. So if we can check over here, if we check all these many of them will be are being rejected, and these the 77 of them have been accepted. So that is why even though it's a small this one, there will be a lot of artifacts still being there. So here we have to update, we have the artifact rejected data set has been created, followed by we have to just update these marks so that we can reject it all the rejected epochs can be removed. So we just remove as a usual step. So here all these channels, so if you go further, there will be various other channels also will be there, which has been remarked as a problem. So I mean, which has been marked as been rejected, so just reject it based on that. So over the so if you see of total 120 epochs, 77 was accepted and 43 was being rejected, and we rejected those 43 trials from the whole data set. So now when we create a new data set, it will be having just the 77 epochs alone. Okay, now what does this 256 actually mean is that for one epoch there will be 256 data points. Similarly, so if there are, so for total 77 epochs, there will be 256 into 77, that many epochs, that many data points will be there. That is the that is our interest. That is why we are using this particular that is the meaning of this particular this frames per epoch means that only. Okay, now next, after ratified rejection, the next step is on filtering the data. Again we use the Butterworth filter similarly with 1 to 30, and then we just do the filtering. So here we have the filtered data set. After doing this particular step, next is the averaging it. So as I have mentioned before, the averaging has been done for since we are working in very few microvolts of data. So what happens is just a single epoch is not at all enough for analyzing it. So we just do the average of the whole 77 to get the proper analyzed ERP. So we just make it as VP. So here we are making the data ERP set. So here ERP set has been created. Okay, now we will just visualize it. Here there is no any bin operation or channel operation because there is just one bin left there. So we just have, if you want, you can do any channel operation, like for a particular way. If you want only the occipital region of the brain alone to be averaged together, then you can just take the bin. A channel operation can be done, but I am just going to show an individual response of how it looks. Here also we take a negative up, or we can take a positive up also, anything is fine. So I just take a topographic analysis of it so that you get a proper. So we have done the ERP lab. Yeah, so this is the proper. So here also we see the reversal taking place. It's a bit less only, but yet there is a reversal. So here it starts itself from the top. From here it's like from down it is going up. So that's like a reversal taking place. And this is the main, this particular region, this occipital area is the main place wherein we get the visual visual evoked potential. We can actually make it a positive up or negative up, anyways is fine. So here if you see, this is how the AEP is looking. It's a bit different, but then as I told you, the amplitude and the latencies are the interest of our that is the point of our interest. So here this is the positive up, the negative up, which is coming in this region. Actually this is a bit weird. I mean, it's a quite a different data set. You can get, yeah, so here we can see a proper N1 P1 peak, the N1 peak and all. So always a P, we can first check is the N1 peak. I think it's better to do a negative up. I did the positive up in the previous thing. I'll do a negative up and check how it looks. Negative up is a bit better because we are mostly checking the negative peaks more, so that's why the negative up is better. So okay, anyway, so this is how the VP looks. Now next I have to make you I have to show is about the how to do the wavelet analysis. So here in this case we can directly, so as I told you, there are 77 epochs and there are 256 data points for each epoch. So what happens is we will have totally 77 into 256, that many number of data points we will be obtaining. Okay, so yeah, this is a positive dip and this is a negative up and again a positive. So this should be the N1 response actually, and this should be the P1 response followed by the yeah, so this is how it should look like. It's not a proper VEP, but this is how it should look like. There is a proper N1 P1 response that has been obtained, so this is how it should look like. Anyways, so I'll just do the exporting now. So we can just go into files and do the exporting of the data. So when we export in this manner, what happens is we can just change the extension. So we just put as Excel sheet, so we can just save it in this manner. We can have how many our digital, how many data points, how many decimal points late afterwards you want, everything you can take it. You can do the transfers, everything that also can be done. So I just save it. So as we save, we will have the VEP being saved in Excel. This is the exported data now. Now if we open this, we will have the whole of for each electrode and for from -50 to 200. So so there are 256. So if you see, there are only 256 samples for one epoch. So similarly there will be for 77 epochs there will be the whole data set. So there will be totally 1 into 117. So these many data points are there for the whole ERP set. So here in this we can just copy the whole of a row, copy it and put in a notepad, and then you can just save it as for example, I'll just save it as FP1 and I'll just put the extension as ASCII format, because always the wavelet analysis, it should be saved in this ASCII format. So that's why we just put in this way. If you want negative up, this is all the positive up data points that has been there. If you want negative up, you have to just multiply the whole of this data sheet with -1, so that you will get the whole of it as a negative up version. So next I'll just show you the data. This is the MATLAB code for the wavelet analysis. So here these three are the main important parameters that you have to check about. So here these are the sample size. So here the sample numbers, which means that for each epoch there are 256 samples. So we will put that as 256 over here, and this particular thing that is called as a sampling rate, and for our case the sampling rate is 1024, so that we enter in that manner. And then this stimulus over here is actually nothing but how far you want the axis to move up and down. So that has been given by this stim parameter over here. Now in our case we have taken from -50 to 200 millisecond. That is the time range we have taken. So just -50, that for that range we have, it should be just this much only, like we have to just divide this by 15 like that. So we will get 51.2. So that we can get a data set from -50 to 200. So I'll just have given this. Then this is nothing but the contour plots and the trial numbers, that is just to, yeah. So I'll just run this particular data set. Even this also we have to do the path, the event, the set path we have to give here also. So we can just download this particular thing we can download from anywhere, I mean in the website actually. So from there we have to then give the in the set path we have to give this particular folder, so that you can just, and then you have to open this and you have to make the changes accordingly, and then you have to run this set, because there are some parameters in this that is required to be opened in this way. So we will just load this particular, I have already made few of this this occipital region electrodes and all. So I have just made some of them, like as I showed now, the ASCII file has been made for those. So I'll just open this OZ. Okay, so if you see here, so this and this, they are both almost the same. Yeah, these two are almost the same. So this is called as wavelet. This is the wavelet software that is being used, and here we have the these are the different the wavelet coefficients, as I mentioned before. So each of this coefficient which will move across all the ERP averages, and that will obtain this particular waveform in this format. So here these are the single trials. For each trial it looks in this manner. So like there are nearly 77 trials, so like this it looks. This is the contour plot that has been obtained for this. So here this is the proper negative, okay, and this is always a negative here, even though it is shown as two, positive up and negative down. Actually I have imported a data which has been a negative up. So this is how, this is actually the positive peak, and positive peak is given as the response is blue response, and this is the red response that has been obtained. Now these are the automatic denoising that has been there, the neighboring as well as the NZT. These are the two advanced denoising algorithms which is actually been mentioned in detail in their Quiroga paper. So when we do this, we will have a particular so it's almost completely overlapping over the system. This red color before, the one which was there, that was the original data set which we given, and after doing the denoising we have obtained the red colored waveform which gives the denoised data set. So here we can clearly see that the blue colors is the positive peak, and the red colors is the negative peak that has been obtained. So basically this will be the N1 response, and this will be the P1 response, and followed by then there will be a P2 response. So here we can get as you move up, sometimes what happens is there will be the coloration will be reducing. So here it is dark and here it will be much lesser, or like that it happens. So which means that there is a habituation occurring. So this is about the wavelet. So here similarly we can create various data sets. So I'll just show you for PZ also. So this is another one ASCII file I have been given, I have been made. So here also we have the we can create the contour plot. So here it is not much prominently seen because it's not so prominently seen in this also. So but after denoising, maybe you will have a proper response being obtained. So see here, this is the negative and this is the positive peak, like that. So this is how the wavelet analysis work with respect to the Quiroga paper they have mentioned. So this is about the wavelet analysis that has been done using this EPDEN software. There are various other parameters which we can change and work about it, like the time maximum, time minimum, the contours, the trials, or even we can change the sampling rate, the sample size. Accordingly, everything can be done. That is about the wavelet analysis and using the VEPs. And then even in this also we can have the similar approach of getting the codes and all for this. So we can just copy paste, add loops, add any other things, and also that for each subject, for all the subjects we can do the similar analysis. Even we can create some codes in MATLAB itself, so that the data, this EEG Excel sheets and all can be converted into ASCII files, column based ASCII files, so that an advanced MATLAB approach. It's like an advanced people who are pro in MATLAB, they can do this type of playing with the numbers like that with MATLAB and all. So that is about the wavelet analysis using the VEP. Thank you.